Hello, my name is Nicholas Espinoza, and I will be explaining the new strategy used to produce natural colorants made with bacteria. For starters, what are natural colorants? Well, naturally produced colorants are dyes derived from plants, minerals, or invertebrates, which is just an organism that doesn't contain a backbone. Because of their natural ingredients that do not contain any synthetic materials, these dyes can be used for items such as cosmetics and clothing, but also food and chemical and pharmaceutical industries. Now, the second question is how they accomplish this. The original strategy is simpler than one would think. They would extract the dyes from plants themselves. This process would start with removing the desired part of the plant that you would want to use for the dye. For example, scientists will use the leaves of plants that belong to the plant genus Indigofera to produce the color indigo. If you didn't know, a genus is essentially a group of species, in this case plants, that are similar to each other, like how the genus Indigofera are similar to each other because they have an indigo component. Anyway, you would then chop pieces such as the leaves, blossoms, etc. for more surface area. After, you would combine them into a large pot and add water to cover the plant material. You would then simmer it for about an hour or until the plant material looks faded and the dye mixed with the water. Finally, remove the excess plant material and the liquid left behind is your dye. This process, however, was labor intensive and came with varying results. I assume this is because if you attempt to industrialize this, then it would be a bit hard for workers to do. So, Sang-Up Lee, a chemical and biomolecular engineer at the Korea Advanced Institute of Science and Technology, planned on using Escherichia coli, or E. coli, a common bacterium, as a new strategy to produce 7 natural dyes, creating 2 never-before-made colors, green and navy blue. The process first started with the scientists inserting genes into the bacterium that would produce the dyes. The problem was that the dyes used were water repellent, so they were not able to exit the cell because of the cell's membrane. The dyes would instead build up inside the cell and eventually kill it. Because of this, scientists had to genetically alter the E. coli. They grew longer E. coli bacteria for extra membrane surface, and used this to create sacs. These sacs were little bags of fluid that would be able to expel the built-up dyes. To create these sacs, instead of replacing genes, which would have a chance of killing the bacteria, they would insert small RNA sequences that would silence the genes they wanted to replace with the sac genes. I'm not relatively sure what process they used regarding the mRNA sequences. I assume, however, it's the process RNA interference due to the use of small sequences. This is a natural process that stops gene expression using non-coding double-stranded RNA genes, and it's through translational or transcriptional repression, so it silences the genes through translation or transcription. They also inserted human genes that would create very small holes, smaller than the sacs, in the membrane that would further expel the dyes. This process helped with the water repellency problem and was ready for industrial use. The only problem with this process, however, was that some colors were produced in smaller amounts and were more difficult to produce, which made them more expensive. So, in conclusion, my biological discipline is bioengineering. My main idea is that scientists are able to use genetically engineered E. coli to produce natural dyes that have no kind of released pollutants like other synthetic dyes. This process has far better results than processes before and is ready to be used for industrial production. My future ratifications are that the method that Lee and his team use can help produce very hard to produce antibiotics, primarily because of their water repellency. Also, Lee wants to be able to make more naturally produced hues using this method. He joked that he wanted to make every compound in the chemist's go-to Sigma Aldrich catalog. If you didn't know, the Sigma Aldrich catalog is just a list of the chemical compounds for different colors.